hospitals for expectant mothers in Kenya. And since 2013, they have given transportation to aid in the births of over 2,000 babies, which is amazing. So how you can help and how you can participate is you can go onto our Instagram, the link is right there, and find the Venmo bingo card. I'll also email that to your exec board after this meeting. And if you could post the Venmo bingo card on social media and any of our other informational graphics that you want to, but that part's optional. And then you can collect the funds on your personal Venmo first, and then screenshot the total amount and then transfer that to our Baylor AMWA Venmo. That would be the way to do it. Or you could also make like a personal donation and you don't have to go through the Venmo bingo card if you wanted to. And so the person who has raised the most money by October 30th will receive a $50 Amazon gift card. So we really hope to see you all participate and raising money for this amazing organization. Thank you for letting me participate, for letting me speak. If you have any questions, don't hesitate to email me or email AMWA Baylor. So thank you.
Great, thank you, Hannah. All right, again, these are all of the burst officers. So if you haven't talked to us yet, feel free to shoot us an email, or if you see us on campus, just stop us and say hello. We'd love to meet you. And then the members of the bi-week, we actually have two this time. So one of them is Cyril and the other one is Caitlin. Are you guys here today?
Yeah, hi.
Yay, hi Cyril, good to see you. So both of these members have just been, outstanding members these past 2 weeks, especially at Research and Internship Day. They were both super engaging and just really interactive with all the speakers. And Caitlin also has been regularly coming to all of the post-meeting socials. I think she's a bit of a post-social or post-meeting social queen. So hopefully she'll be there today as well. And Ruhi will be emailing you guys so that you can get your free t-shirt. And then more exciting news, Student Activities has actually approved the start of in-person organization meetings. So BURST is going to start cautiously hosting a limited number of in-person meetings in the coming weeks. So just be on the lookout for those in the weekly email.
Just a quick reminder that a minimum of 16 points needs to be earned by the end of the semester to remain an active BURST member for next semester. And so for most of you, the special event point has already been completed from Research and Internship Day. So most of you just have to focus on the 7 general event points and the 4 journal club and service event points. And if you're also a part of a research lab, you're welcome to fill out the form that's linked in the slide to earn 6 points of research credit, which will be accounted for at the end of, or close to the end of the semester. And regarding membership and dues, for those of you that haven't paid your dues yet, the fee is $15 for returning members and $25 for new members. And for new members, you also have to fill out the new member form, and all dues can be paid via PayPal at Baylor Burst or through our Gmail.
Okay, so next week, we have an exciting engineering and computer science guest lecture event coming up. Burst actually partnered with the Society of Plastics Engineers to host 2 Baylor alumni to come back and speak about their research experience at the Southwest Research Institute. So if you're interested in attending, it'll be over Zoom next Thursday at 6:30 p.m.
Here are the next week's following journal clubs coming up. There's still a physics club tomorrow if you want to. The sign up was sent in last week's weekly email, or you can fill out this form at the bottom of this slide.
And then, so these are the socials for the coming week. The Burst Scavenger Hunt was actually yesterday evening. So shout out to Juan and Sean's family group. They were the winning group. And shout out to the members, Luke and Haley. You guys will win a special prize. So actually we'll be getting that to you guys soon. Our third research coffee hour is next Wednesday, and it'll be with Dr. Doyle. He's a professor in the biology department. And then right after this general meeting, we'll have our post-meeting social. We're going to be playing Among Us again. So definitely, stick around and get to talk to your fellow burst members. And if you haven't been assigned to a family group yet and you want to, go ahead and just email Akshat and he'll get you assigned.
Alrighty, and then Scientia is open and it'll be open all the way until the end of December. And we accept anything from original research papers, review articles, and abstracts. So if you have done any undergraduate research or know anyone who has, and they have something like this, definitely send them our way.
Okay, so in the fall, the Scientia Committee works on our research newsletter, and so it's going to be released on November 2nd, so pretty soon. You can look forward to a lot of information on upcoming internship programs, an interview with Baylor College of Medicine's Dr. El Sally on the Moderna COVID vaccine, and a lot of testimonials and interviews from really cool Baylor student researchers.
All right, so for academic workshops for October and November, so on the 26th, this upcoming Monday, we'll be inviting Representative Mr. Haynes of the Career Center, the same one from the resume workshop, to talk about the process and the resources for graduate school admissions. And so that will be around 4:30 p.m. until 5:30. And for lab techniques, this will be the first time we're hosting virtually on Zoom, and this will be on the 28th. And so this is on a Wednesday, and we'll be introducing to you all the instruments that you'll be using for your upcoming lab courses and research labs. And so the third one that we have for you guys, early November, is our first ever LinkedIn workshop. I'm really excited about this one. So this will be on the 9th of November. That's correct. Yeah, at 5 p.m. And so LinkedIn is a great resource for improving professionalism and networking, and even putting out your name for possible employers and connections. And so whether if you have a LinkedIn or not right now, or you'd like to improve your profile or get started on a new one, we'll be here to give you all the tips and resources and answer any questions you have. And lastly, for mid-November, we have our alumni, Emily and Zach, coming back here on Zoom, of course, talking about their experience with applying to graduate schools, what they did over undergraduate years. And they've done a lot for bursts during their undergraduate years, so they'll be really helpful. That'll be at 4 p.m. on the 12th. And so, without further ado, we'll be introducing our guest speaker for today's meeting. I'm really excited to have Dr. Greathouse talk with us tonight. And Dr. Greathouse specializes in consumer and nutritional sciences and cancer biology. She has done amazing research on applications on diet and metabolism, as well as incorporated bioinformatics and machine learning tools to help with the studies. And Dr. Greathouse has published numerous papers, and you all will be able to check them all out on the Baylor page. And we can also provide a link for you all as well. And so Dr. Greathouse will be sharing about her background and current and future research. And so without further ado, I will now hand it off to Dr. Greathouse. Thank you.
Thank you. So let me share my screen. All right, there we go. Can everybody see my screen okay? Perfect. All right. Well, I was really excited about your introduction. It reminded me, I sat down and wrote like a bunch of things I wanted to tell you before I get started. I don't have children, so I like to impart my knowledge on people of your age. So one of the things I wanted to ask you, maybe you can do a Q&A while I'm talking, is what is your favorite movie? Because I have all of my favorite nerd friends right now, I think, surrounding me. So my favorite movie is Real Genius. It is an amazing 80s B movie, and it's fantastic. I love it. I watch it probably like once a year. All right. My favorite book, well, for networking, is called The Third Door. Totally worth the read. Y'all will really like it. My favorite book for making good arguments is Love Your Enemy by Arthur C. Brooks. And my favorite nerdy book right now is The Perfect Predator, about one of the scientists who discovered phage therapy, or reinvigorated phage therapy for her husband, who is also a scientist, to help him recover from an Acinetobacter baumannii infection that was killing him at the time. So 3 great books. but I'd love to hear what your favorite movies are and your favorite books are. So post it in the chat if you want to. All right, so I will get started with my little background information for you guys. So I started out life as a dietitian. You might not know that. And during this time, I thought, I wanted to help people eat better. I was a big fan of eating healthy. I always had been. I'd been an athlete my entire life. And so eating healthy was a big part of my life. And during this time, I thought, well, maybe I'm going to take the next step and become a sports dietitian, kind of, which fit with, my background in sports and gymnastics. And it was during this time I was doing my master's degree at Texas Women's University. And because they were one of 6 places in the United States at the time, back in 1998, that had a sports nutrition master's program where you could get your dietetic degree and a sports nutrition degree. And during this time, I was diagnosed with a really rare form of cancer. It was called a uterine leiomyosarcoma. It was a stage 4, so they didn't give me a whole lot of hope. But I had a really great team of doctors and support team, and my church with family was really supportive. And so I did 6 rounds of doxorubicin, and by the end of that year, I had a clean scan. It was amazing. So that really catapulted my confidence and changing my career trajectory and getting a PhD in cancer biology. So you can imagine going from a dietetics degree to cancer biology is quite a shift. So it took a big leap of faith to actually do that. After I finished all my treatments, I applied to some of the different doctoral programs. Mainly I stayed in the Texas area because I actually got married at the time. And so I looked at places in Texas, and University of Texas is one of the places that accepted me. And that's a really great program because it has both the Graduate School of Biomedical Sciences at UT, and which is associated with MD Anderson. So it's a really great place to get a degree in cancer biology. And so my degree actually was officially cancer carcinogenesis. And actually Dr. Taube, who you might know here at Baylor University, we graduated from the same program around about the same time. So we're both cancer biologists. So during this time, I became really fascinated with the microbiome because to me, it married the best of both things, that the diet affects the microbiome, which are all the bugs that live in and around your body and mainly in your gut. And cancer is influenced by your microbiome. And I thought, what a great way to look into ways to prevention and treatment of cancer than 2 of my most favorite things to think about, the diet and cancer, is looking at the microbiome. So I was really fortunate that one of my mentors during my PhD, Steve Hursting, was actually the former director of the cancer prevention program at the National Cancer Institute. So he suggested, based on my interest in science policy at the time, I had a brief interlude of being a science policy analyst, but that waned. So he suggested that I go to the NCI and do the Cancer Prevention Fellowship. Little did I know how competitive this Cancer Prevention Fellowship was. I mean, you actually get paid really good money right off the bat. It's usually you get paid about $48,000 a year for a postdoc. This was starting out $65,000. So it was really great. And also very competitive, but thankfully I had an ace in my back pocket, which was Steve Hursting. And that I think really helped me get into the Cancer Prevention Fellowship. So the Cancer Prevention Fellowship is really unique in that from any other postdoc, you get training and learning how to speak to people, give presentations. You get training in grant writing, which is critical, at least for somebody like me who's an academician, survives on grants. And you get to pick from any of the researchers at the National Cancer Institute to be your mentor, as long as they accept you, because you're free to them, because they don't have to pay for you, which is really great. So the Cancer Prevention Fellowship was a really great catapult for my career. Oh, and the best thing, I didn't even tell you, I got a free master's in public health from Johns Hopkins School of Public Health, the #1 school in the US, because I was in the Cancer Prevention Fellowship. So thank you, taxpayers. I appreciate it. So I spent the next several years training myself how to do microbiome analysis, because I wasn't a microbiologist. I had to train myself. I literally cold called people around the NCI that would take me, and thankfully they did, into their lab to train me. And so I put together a team to actually analyze microbiome data. So that's what I'm going to talk to you about for the rest of my talk today. But in this time, I taught myself how to code in R mainly. So if nothing else you get from this talk today, learn how to code, period. You'll have a job for life. All right, let me get started. With all of this information that I gathered and all the training I have, I'm actually now qualified as an expert poop collector. And that's what I have to give to y'all today, is how to collect poop. Hopefully it's more than that. So my team I call Cancer Crusaders, and these are the rest of my team members. These are my fellow Cancer Crusaders, and they are the main people who make this engine work. And I'll be showing you data from some of their work today as well. And I have a huge cadre of undergraduate researchers that I love to mentor, because that's one of my favorite things to do, is mentor undergraduates and graduate students as well. So this is the outline that I'm going to be using today. I'm going to talk to you a little bit about the driving question that drives most of my work. I'm going to tell you about my main work, which was the lung cancer microbiome, which kind of started out my career, and then kind of go into the future directions of where we're headed right now, looking at the microbiome. So the driving question behind most of my research. So I don't know if you've ever seen one of these little charts before. It's an epidemiological model, and it shows interactions. The main ones that we know about, I'm sure you're very familiar with, is obesity and cancer, which drives cancer risk. Diet and cancer is a strong connection. And this PAF is population attributable fraction. So there's a 6 to 9% PAF for cancer from diet, but obesity drives cancer even more than diet does. However, what we don't know, and which is the gap that I'm trying to fill, is the microbiome. So that's the main focus of my lab, is looking at how does diet affect the microbiome, and then how does that influence cancer development and treatment response. So overall, if you were to look at microbes in general and their contribution to cancer, it's about 15 to 20% based on which article you read. And the main ones that we know that actually initiate cancer, meaning that they are sufficient in and of themselves to start cancer, are things like HPV, HBV, and H. pylori. And we know, or hopefully you'll know, that HPV, there's a vaccine now for that called Gardasil, which is great. But the microbiome has many other factors that influence it besides diet and obesity. Geography is a huge component of what governs the microbiome, as well as the immune system. So many different things. For some reason, I can't move forward. Let's see if that works. No, still can't move forward. Let's try this. There we go. All right. So the things I'm going to be focusing on during this talk mainly are these information right here, but many different things happen that affect the microbiome. Mainly there's, if you look at the first picture here and under A, there's many different bacteria that we know that affect the microbiome, and they actually affect cancer as well. So F nucleatum is one that's really found in colon cancer. I just talked about some of the viruses that also affect inflammation. Some of these bacteria also affect the barrier integrity of the colon. And some of the ones I'm going to be focusing on later on are these bacteria that actually cause inflammation through certain pathways. This is one of my absolute favorite pictures because it really explains the intimate relationship between the bacteria and our epithelial cells and the host immune system. So what this is explaining is that if you look in the colon, there are really two important key barriers that don't allow bacteria to touch our epithelial cells. And this is important because you don't want them in direct contact because that causes inflammation. So we have this thin mucus barrier here that keeps the microbes intact, and this thicker mucus barrier here that really prevents, it's kind of like a demilitarized zone, preventing the microbes from touching the epithelial cells. So one of the things that's really important is fiber in our diet. My other thing, take home message today, learn to code and eat more fiber. So fiber really affects the microbiome because a lot of them really have the ability to degrade fiber in our diet. And they use this to produce a really important molecule called butyrate. It's a short chain fatty acid that's critical to keep our colon epithelial cells really healthy. And what this also does as well is it suppresses the pro-inflammatory immune factors. So it dampens down that hyperinflammation. However, if you eat a diet that is very low in fiber, like fast food all the time, processed food all the time, then what happens is these bacteria that normally forage on the fiber that's in your diet will now start foraging on the carbohydrates, the mucins in your mucus barrier. And so now they eat down this mucus barrier, and now you have intimate relationship, which is not good, with your microbes leaking across, decreasing the barrier function, and increasing the inflammatory response, leading to probably chronic or potentially chronic inflammation. So this is what I study and try to understand in my research is how does this chronic inflammation occur from the microbiome? So in the normal microbiome, at least in the colon, you have usually a high diversity, you have pathogen clearance and immunoregulation. You have good barrier function, balanced cell death proliferation, and anti-inflammatory response. Now, when you have colon cancer, this bacterial diversity decreases because certain pathogens take over. I kind of think of it like the free market system. You don't want to have monopolies because that's not really good for anyone. So you have this microbiome imbalance, you have immune evasion, you have disease in the host, altered or disrupted barrier function, and inflammation. Now, if you look at this as opposed to colon cancer and you look at lung cancer, this is what I studied during my postdoc. Again, you have low diversity in the lung because you have this mucociliary escalator that beats and keeps the microbes out. And you have the healthy host, but in the lung cancer microbiome, this mucociliary escalator is no longer keeping those microbes out. And now you have an accumulation of microbes, which increases the diversity, which is not actually a good thing. So this is actually what I studied during my postdoc. So some of the things that led me to investigate this further were these important pieces of information that I identified in the beginning. So that we know that chronic inflammation precedes multiple different types of cancer. There's in fact, tobacco smoke can induce epithelial dysfunction. We know that chronic obstructive pulmonary disease is a risk factor of lung cancer and is associated with increased bacterial colonization. We also see that lung cancer development is reduced in germ-free mice infected with pneumonia. And this important component, LPS, a lipopolysaccharide, increases inflammation in lung tumorigenesis. Now, this information that I'm showing you down here is really important to the rest of my talk. So let me explain what I'm showing you. So in mice that have a specific tumor suppressor gene called P53 deleted from only their intestinal epithelial cells, they have migration of bacteria across the lumen into the cells below. And what this causes is an increase in inflammation, because this tumor suppressor gene not only suppresses proliferation, it also tightens up those barriers so you don't have microbes tracking through. So that'll become important later on in my talk. So just an example of how do you do microbiome analysis. And we'll come back to this a little bit later. So usually what you do is you can get saliva from people, or you can get stool samples from people. In our case, we actually got tissue samples from the lungs of people. And later on in my other work, I'll talk about colon. And so what you can do with this is you can do DNA sequencing. And a lot of people do what we call 16S sequencing for microbes just themselves, or you can do shotgun sequencing, which sequences everything that's there. And there's bioinformatic tools called Human, Metaflon. There's any number of bioinformatic tools that you can do. Again, learn to code. Okay, and then there's my little publication that I had. Okay, so this is an example of what we had to do to gather all the information to do this lung microbiome study. So we had two populations. When you're doing biomarker analysis studies, you need a discovery cohort and a validation cohort. It's critically important. So we had a discovery cohort. This was a 30-year long-standing cohort in my PI's lab at the NCI for lung cancer. And we had several hundred frozen tissues that we used to do DNA extraction. I also had 2 non-cancer tissue controls and 2 negative controls. And this is absolutely critically important. You're dealing with very low biomass samples. And so you can imagine that any microbe that traces into your samples that was not originally supposed to be there is going to be amplified when you go and do sequencing. So we had to be very careful when we did all of this analysis to really look into what were the background contaminants for each of these types of samples. So we had 2 different methods of sequencing. Again, we did 16S sequencing for just the bacteria that was there. And then when we had the bacteria that we identified, we did longer reads called PacBio sequencing to be able to identify the strain of bacteria. We also did targeted sequencing for that gene, that tumor suppressor gene I mentioned. And then FISH is just fluorescent in situ hybridization. And we also did strain identification. Our second validation cohort was from the Cancer Genome Atlas. I'm not going to get into some of the papers that were recently published from this study, not mine, but somebody else's. Suffice it to say, I am not a fan of what they did with the microbiome analysis in that. So we did, but the reason we used it is because it had over 1,000 tumor and non-tumor samples in it, and it had data on P53. So that's overall the study that we used. And so what I did was we did a relative abundance analysis, and what you're seeing here is a heat map between 2 different types of lung cancer, adenocarcinoma and squamous cell carcinoma. So what we identified was several bacteria that seemed to be higher in abundance in one type of lung cancer versus another. One of these was called Acidovorax, and the other one was Klebsiella. And what we did was we looked in our TCGA study to confirm this using more sophisticated analyses. And we were able to conclude that there are several species of Klebsiella and Acidovorax that seem to be highly related to squamous cell carcinoma. So one of the other things I didn't tell you about squamous cell carcinoma is, of the lung cancers, it's the one that's most strongly correlated with smoking. So the next thing I asked myself, well, if this is truly correlated with smoking, I should see some of these bacteria that are just purely associated with smoking or not smoking. So I did this fancy analysis called linear discriminant analysis. And the bacteria that came up as most strongly associated with smoking was, one of them, of course, was Acidovorax. And so I went back to my NCI cohort and I asked the question, well, if I were to further define smoking into never, former, or current, I would expect to see more Acidovorax in people who were current smokers, which is what I saw. The next thing I asked myself, well, if this was truly associated with smokers or former smokers and squamous cell carcinoma, I would expect to see more Acidovorax in those samples. And in fact, that's what I saw. So in squamous cell carcinoma, in the NCI study and my validation cohort, I saw more Acidovorax in both of these samples, in both former and current smokers. And so one of the things that you absolutely have to do in these low biomass studies beyond sequencing is do some type of other method. And here I'm showing fluorescent in situ hybridization for probes that are specific to Acidovorax. And so these little tiny red dots are those little bacteria that are associated with Acidovorax. And so we took a patient samples and we counted the number of cells that were positive in those tumors for Acidovorax. And the last thing that we did for the study was look at this association with P53. The other thing I want to tell you is that in squamous cell carcinoma, the top mutation in 70 to 80% of all squamous cell carcinoma tumors is P53. And so I asked, we know that P53 is critical for shoring up that barrier to prevent encroachment of bacteria in. And so the hypothesis would be that if we had a deletion of P53 or loss of P53, that you would have more bacteria in that tumor. And that's what we see. We see people as tumors with P53 mutations versus wild type had much more Acidovorax in their tumors. Now, this is not causation. I'm not saying that Acidovorax is causing lung tumors, but I'm saying it's associated. We can't say it's causative unless we do more in vitro work and in vivo work, which I'll show you later. So just to show that this is also validated in other studies, 2 other papers, and there's actually 3 now that have shown the same association with Acidovorax and lung cancer development. So my colleagues at the NCI continue to do this research after I left and ask again this question, does Acidovorax play a functional role in development of lung cancer? So this work was done by Sharinin and Dr. Harris, my former PI, and they used a mouse model of lung adenocarcinoma, and they inoculated this mouse with a type of adenovirus that would delete these genes, specifically in the lung, and lead to lung tumor development. And so then we asked if these bacteria, either Acidovorax temperans or a commensal algaceri, would actually increase or promote lung cancer. And in fact, when you look at that, it does lead to lung cancer promotion. So these are MRIs of the little mouse lungs at 13 weeks. And after they were challenged with Acidovorax, and you can see the tumor volume is much higher in those challenged with Acidovorax temperans, indicating that indeed it does promote lung cancer. Not that I'm saying it's sufficient by itself to start lung cancer, but it's a cancer, potentially a cancer promoter. My other colleague, Josh Stone at the NCI, further went on to look and see if these pro-inflammatory cytokines are correlated with bacterial abundance in the TCGA samples. And so you can see in the green versus the purple here, the squamous cell carcinomas, that there's several, including IL-8, and that's associated with inflammation. So what he did was he now looked at this expression of IL-8 in correlation with either Acidovorax or Klebsiella. And you can see by the p-value, there's a very strong correlation with Klebsiella in adenocarcinoma, but even a stronger correlation between IL-8, Acidovorax, or Klebsiella and squamous cell carcinoma, indicating that this is a strong relationship between these two bacteria. One of the other things we did is we took this gene signature that was associated with what we call T-cell exhaustion, meaning that these T cells have been bombarded with tumor antigens so long that they're now just pooped out and they don't respond like normal. And there's gene signatures that we can identify that show this kind of analysis. So he looked again at this in the adenocarcinoma, which was not really significant, but in squamous cell carcinoma, it was highly significant, indicating that there was a T-cell exhaustion signature there in squamous cell carcinoma. And when we compared this, again, with the squamous cell carcinoma, looking at these T-cell exhaustion signature, it was highly prevalent here, as these red squares indicate, that there was some type of T-cell exhaustion going on in squamous cell carcinoma. So then we asked the question, well, is this correlated with these bacteria? And so you can see that in adeno, and even more so in squamous cell carcinoma, that this T-cell exhaustion is actually correlated with this signature and with these bacteria, indicating that one of the potential ways that tumor promotion is occurring is that you've exhausted the T cells, you've hammered them so hard for so long, they're no longer responding, and now these bacteria are taking over and promoting lung cancer. All right, so what did we identify? A group of taxa that are rich in adenocarcinoma, and mainly in squamous cell carcinoma, the Acidovorax genus that's associated with P53, and Acidovorax temperans that can increase lung cancer in these animal models, and the cytokines that are correlated with squamous cell carcinoma. And finally, Acidovorax and Klebsiella that are also correlated with T-cell exhaustion. All right. But the question that I am trying to answer with some of the data that we are doing in our lab currently is, how are these microbes promoting inflammation? What is the mechanism? So this is actually one of the talks that my doctoral student Adil gave today. And so I'll just walk you through. I talked to you a little bit about lung cancer, and now I'm going to talk to you about colon cancer. So just like lung cancer, colon cancer is associated with inflammation, and the microbiome is a significant contributor to that inflammation. And one of the bacteria that's been significantly associated with development of colon cancer is one of them called Bacteroides fragilis. And the cool thing about Bacteroides fragilis is that it exists as both a commensal and a pathogen, and it can have very distinct outcomes in health, either as a promoter or as a promoter of microbial tolerance or inflammation, depending on the context in which it lives. And so the way this works is, right now that we understand, are these microbes can interact with surface receptors on our cells called toll-like receptors, and they can promote microbial tolerance. However, the pathogenic form actually produces a toxin that can disrupt this tolerance. But one of the other things that we're investigating is whether or not these little vesicles that bacteria secrete can actually activate toll-like receptors and further increase inflammation. So one of the ways that we think that inflammation is being increased from these bacteria are through outer membrane vesicles that all bacteria secrete. And so these outer membrane vesicles host a range of different cargo, nucleic acids, toxins, metabolites, they use them to communicate with each other. And so we hypothesize, well, if they communicate with each other with these vesicles, perhaps they communicate with us with these vesicles. And so that's one of the things that we're testing in these analyses. So the way that our cells recognize all different types of microbes are through pattern recognition receptors called toll-like receptors. Now, there's many different kinds. Some of them live at the surface of the cell and they recognize bacterial products. Some of them live inside the cell, inside the endosomes. And these recognize nucleic acids. And those are the ones that we're most interested in, as the one that recognizes nucleic acids, because we believe that bacteria secrete small RNAs that could potentially activate these. They increase this pro-inflammatory cascade and the adaptive and innate immune system as well. So our hypothesis is that these pathogenic versus commensal strains have OMVs they shed with distinct cargo that can engage the toll-like receptors on either the surface or the endosomes and create either a pro-inflammatory cascade or an anti-inflammatory cascade. So I'm going to skip this because I don't think you care about vesicle isolation. But what I will say is that the way that we use to analyze these vesicles was a toll-like receptor or toll-like receptor reporter cells. So it's hooked up to a really cool promoter that expresses a protein that changes color that you can look at under the microscope. So this is what we did to test whether or not these OMVs were actually activating inflammation. And so our first activity we looked at was toll-like receptor 4, which recognizes LPS, which is a product that bacteria secrete. And we do see, in fact, that both NTBF and ETBF respond to this as we expected, and this is our positive control. And then we next ask, well, if RNA or nucleic acids were actually important, we should see a difference in the OMV activation. And so we do see that NTBF actually is strongly activating TLR3, which can bind nucleic acids, and our positive control here is worked as well. And so the next thing we asked, well, if this is recognizing nucleic acids through this TLR3, then we should see maybe differences in RNA between the cargo of the OMV from either ETBF or NTBF. And so we isolated the OMVs and their small RNAs from both strains, and we applied it to RNA sequencing. And so we aligned these RNA reads to the genome of ETBF or NTBF and to see what genes are expressed. And so NTBF, we see expressing a certain set of genes, and ETBF. But there's a significant difference between what RNA species are being expressed in the OMVs versus the whole cell lysate. So indicating that OMVs uniquely package small RNAs. And we dug even deeper to see if there's a difference between ETBF and NTBF OMVs. And we see that there's a small set of genes that are only expressed as cargo in ETBF OMVs versus NTBF. So then we next asked, do these OMVs actually affect colon epithelial cells? So we looked at cancer cells, which are KCO2 cells, using qPCR. So we looked at the inflammatory cytokines, IL-8. We definitely see a response from NTBF, which is a little bit stronger. We also looked at another inflammatory cytokine, IL-1. We see a stronger effect with ETBF. And then we looked at an anti-inflammatory product, TGF-beta. And we see, interestingly enough, that NTBF at different doses enhances the expression of this anti-inflammatory product, TGF-beta, indicating that perhaps our hypothesis is correct. The last thing that we've done so far in this work is to see, well, if we look under the microscope, do we see these OMBs localizing with the endosomes? So we've labeled the endosomes red and we've labeled the OMBs green. And so when they overlap, they should be yellow. And so that's what we're seeing here is these little yellow pieces here and here that indicate, in fact, they are overlapping, giving us our first clue that they are actually interacting. So you still have a long way to go with this information, but at least what this gives us right now is indication that their OMBs are contributing to this differential small RNA communication with the host, and that is activating the TLRs. And so the next things that we're going to do with this is we're going to use 3D collonoid organ models to get a better mimic of the intestinal epithelial structure. And we're going to use fluorescently tagged OMBs to look at trafficking using live cell imaging. And then we're going to further do more in silico bioinformatic analysis to identify any of these small RNAs that could actually bind to host targets. All right, and so I'll end with my future directions. And this is, I think, what you saw on the Baylor webpage is this new project that we have going on, which is looking at the microbiome as a predictor of response to colon cancer therapy. So as I told you before, there is this understanding that the microbiome contributes to cancer, but what we yet know is how much does diet contribute to this relationship, and that's what one of the things that this project is answering. So one of the coolest things that I love to show people is this study. It was only 2 weeks. They did a diet swap with Africans from rural Africa and African Americans from America. And they did a 2-week diet swap. So they ate each other's diet for 2 weeks and then switched back. Now I want you to look and see what was the difference between the diet. Fiber, right? Huge difference in fiber. 66 grams of fiber. Now recall what I told you earlier. That happens when you eat plenty of fiber. It prevents inflammation, right? And when you don't eat enough fiber, all that mucus is degraded and leaves your epithelial cells open for inflammation. So in 2 weeks, just by doing the dietary exchange, the African Americans, the men, they had decreases in inflammatory response markers. They had changes in their microbiome and decreases in metabolites that were markers of colon cancer just in 2 weeks. So this information was really important. I don't know what happened to my slide there, but this information was really important in helping get this information off the ground and the study off the ground. And later on, what we saw was that in a study where they were looking at melanoma and response to immunotherapy, they saw that people who were eating higher amounts of fruits and vegetables, whole grains, high fiber diet had a better response to immunotherapy, okay? Diets with higher added sugars and processed meats were negatively associated with these bacteria. And the most important thing, patients who consumed a high fiber diet were 5 times as likely to respond to immunotherapy as those who consumed a low fiber diet, indicating that yes, response to therapy is really important with relationship to your diet. And so with this rationale, that's what led us to do this study, looking at the diet and microbiome as predictors for chemotherapy-induced diarrhea, which happens in about 50% of patients undergoing chemotherapy for treating colon cancer. And unfortunately, there's no clinical biomarkers for chemotherapy-induced diarrhea. Current strategies for actually predicting or treating it has not worked very well. So that's one of the major reasons that we're doing this analysis, is to identify what those markers could be and maybe be able to treat it later on. So this is just an analysis that somebody had done previously, showing that there in fact is a difference in the microbiome over time, people who are doing radiation therapy, and changes in their microbiome in relationship to development of diarrhea. So this is our study that we have. We're going to start hopefully in the next few weeks, and we're going to be enrolling over 120 stage 2 and 3 colon cancer patients. We're going to be getting stool and blood from them at colonoscopy, and then tissue during surgery. And then we're going to be getting blood and stool samples again before they start treatment, and then before and after treatment on 3 different time points during treatment. And then we're going to be following them for up to 5 years to look at this. So overall, the ultimate goal for this, at least for me, is to identify what those tipping points are in the microbiome that either promote carcinogenesis or predict response to treatment. So that is the main goal of all of this research. And I will just like to say thank you to everybody in my lab. They are absolutely critical in doing this work, and all of my collaborators, and of course my wonderful funding sources. And if you want to, you can follow me at Cancer Microbes. And I will be happy to take any questions.
There are a few questions in the chat, Dr. Greathouse, if you want to look at those, or I can read them if you want.
Okay, let's see here. So it looks like some doctors have said that fiber pills aren't as effective as natural fiber from your diet. If so, do you know how much less effective they are? Would fiber pills still be a good supplement to top a healthy diet? Food is always best. Absolutely. Fiber is not going to replace normal food, but I have no problem if you want to take a fiber supplement. I like the fiber supplement called Prebiotin. It's really nice. It's tasty, and it dissolves in most liquids. And so if you want to add a fiber supplement to your diet, I highly recommend it. Can't go wrong. It's not going to be bad for you. What I don't like is this whole silliness around taking probiotics. Now, if you want to eat probiotics in your food, like kombucha or kimchi or yogurt or kefir, that's great. But you don't need a daily probiotic. That's just silly. There's no reason to do that. There's no data showing that it's effective, and you're just wasting your money, just like taking multivitamins. It'll give you expensive pee. Probiotics will give you expensive poop. All right. From the first study, were you able to correlate smoking with lower mucus production in the lungs? Oh, that's a great question. No, we have not looked at that, but I really like that idea. I will have to talk to one of my colleagues about that. I don't know. It's possible, entirely, yes. I don't know. Those are the only questions I see. Are there other questions? All right. Well, I don't see any other questions. So either y'all really loved it or it was uninteresting. Oh, what got you interested in this field? Well, the microbiome. Well, oh, great question. So the main thing that got me interested in this field was a study from Peter Turnbaugh's group, and they looked at twins. One of the twins was obese and the other twin was lean. And they took stool from the twins and they inoculated mice that didn't have a microbiome, and they fed them the exact same diet. Can you guess what happened to one of those animals and not the other one? The mouse that got the stool from the obese twin became obese. And the stool from the lean, they stayed lean on the same diet. They were eating the same calories. So that means that your microbiome can totally change how your body processes food and energy. That is what got me started into the microbiome. All right. Is there a certain course or online series that you use for learning how to code in R besides R programming language? Okay. Yes, there is a wonderful course from, well, you can do it online at Coursera. Coursera offers really great free courses for online coding. That's one. And there's one from my colleague, Pat Schloss. It's called Minimal R. And I can send that around in an email after we're done here. The other language I highly recommend is Python for biomedical sciences. So R and Python are probably your best languages. Okay, from the presentation, all of this seemed like a very smooth process. Did you run into issues along the way throughout your research? Heck yeah. Oh my gosh. I was teaching myself how to do this on the fly. You know, I messed up a million times trying to isolate DNA before I realized that there was mass amounts of contamination. I had to do that multiple times. I had sequencing fail on me constantly because my sequencing core didn't know what the heck they were doing, because this is the first time I ever done 16S sequencing. So we had to work through that together. That was a problem. What else? Man, coordinating all these people to do microbiome research at the same time, finding a bioinformatician that knew how to do microbiome research. I had to hire somebody outside that I didn't know. Thankfully, he was really great. Processing all of these reads. I didn't know how to do that. I wasn't a bioinformatician. Thankfully, we had a supercomputer at the NCI, but I had to like beg, borrow, and plead for massive amounts of storage space to do all this information, this bioinformatics. Just actually trying to get this stupid paper published was crazy, because nobody believed me that there were this microbe population in the lungs and that was related to lung cancer. It took me 4 times to get this published, 4 different journals. And finally I got it published. But I had to jump through every single hoop to show that this was not microbial contamination and this was real. So yeah, there was a lot of hurdles getting this published. What is the difference between 16S sequencing versus other kinds, and why do you choose that? Well, at the time, 16S sequencing was the best thing that we could do that was not ridiculously expensive to get an idea of who's there, like just the relative abundance. And I had colleagues that were doing that recommended that I do 16S sequencing. Later on, we did PacBio sequencing, which was full length 16S sequencing. And then after that, we did shotgun sequencing. So it kind of, we progressed through the different sequencing modalities. But we started out with 16S because at the time, that's what everybody was doing. Yeah. All right. Any other questions? All right. Well, thanks for inviting me, you guys. I hope it was informative and fun.
Yes, absolutely. Thank you so much, Dr. Greathouse, for being here tonight.
Sure, absolutely. Well, I will email you those other 2 links for coding in R, and hope you find them of interest and learn to code and eat more fiber.
Yes, will do. Thank you.
All right, thanks. Good night.
All right, let me. All right, guys, so don't forget to connect with us. Today's recording of Dr. Greathouse's speech will be on our YouTube later tonight. Follow us on Instagram and Twitter to make sure you're hearing about all of our events. And then check out our website if you need more information about Burst or how to pay your dues. And then thank you guys so much for being here. Here's the QR code. Go ahead, take a picture of it, sign in so that you can get your one general event point. And then if you're able, stay for the post-meeting social. It'll be on the same Zoom link and we'll play Among Us. But that's all. Thank you guys for being here and we'll see you next time.